afternoon, everyone. Hope you had your lunch already, because we would like to invite our second virtual speaker, Dr. Veeranjaneya Reddy, a lecturer from the Department of Microbiology, who joined us directly from Yogi Vemana University in the Zoom platform. A little about him. He is currently a lecturer at Yogi Vemana University. In 2001, he graduated from Periyar University, Salem, Tamil Nadu in Master of Biochemistry. He received his PhD in microbial biochemistry, fermentation technology from Sri Venkateswara University, Tirupati. He has published close to 100 publications, including journals and chapters of books, and received many awards from national and international professional bodies. His field of expertise include the production and characterization of wine from tropical fruits like mango and guava, and production of industrial, chemical, and biofuels using agriculture waste. Now, I would like to call upon Dr. Veeranjaneya to deliver his speech. Please welcome.
Thank you for your short introduction. And good afternoon, and thank you very much for inviting me for this annual conference. And uh, this year, the theme of the conference is agriculture reliance for food security. And with the basis of that, I have selected my talk on agricultural crop residue as a renewable and economical bioresources for biofuels, and why this is going to be a related topic to the food security. And we discussed that. So, due to the increasing population throughout the world, the demand for the energy is going to be increasing throughout the globe. And most of the sectors, the different types of the sectors are using the energy in the form of either electricity or crude oil or some other way. And because of this, we need to uh, given a good importance to the energy research, and also we are going to be demanding for more and more amount of energy to the human welfare and growth of development of the human being and luxury living. And this currently the main energy source of our system is fossil fuels, and these are non-renewable sources, and which are going to be, you might have known well that natural gas, oil, and coal, and these are having using for the production of the fuel, electricity, and other goods. And due to the because of the increasing demand, due to the urbanization, and also the population increment day by day, and the uncontrolled dependency and usage of these fossil fuels, which are non-renewable. And whenever, if you want to generate, these are going to be produced from the renewable sources like plants and other, when they have buried inside the soil, but it takes some thousands of the years to produce these fossil fuels like the natural gas and the oils. And these are going to be expected to deploy the reserves by 2050. It is very near, only 25 years. And because of that, we need to think of the alternative energy sources to substitute these fossil fuels, and also give the alternative support for our demand. And then The increased dependency or utilization of these fossil fuels is causing whatever increasing the greenhouse gas emissions and increasing the prices of the crude oil throughout the world, and also decreasing the long-term availability, and it is also going to be causing many environmental problems, climate change, due to the emissions that are going to be released during burning or utilization of these fossil fuels. Okay, and then we need to, as I already told you, we need to switch to the alternative and also the sustainable fuels like solar energy and wind energy, and also the tidal energy using the water, hydro energy, and then the biomass, and also the geothermal heat wherever it is available. And coming to the biofuels, which are going to be the somewhat different from the fossil fuels or non-renewable fuels, these are renewable. That means these are generating through the plant system or waste systems wherever these are going to be available. And these biofuels, which are going to be producing from the feedstocks, which are less expensive and also less environmental pollution. They are going to decrease the both price and also carbon dioxide and NOx emissions. When you come to these biofuels, we can divide them into the three different types. Here you can see very elaborately and also a pictorial manner that biofuels, we can divide them into the primary biofuels and secondary biofuels. Then in secondary biofuels, you can divide them into the first generation biofuels, second generation biofuels, and third generation biofuels. And in primary biofuels, these biofuels are unprocessed biofuels. So whatever the plant waste or plant materials which are going to be using as it is as heating or for cooking or for electricity generation. So you are not going to be process it anymore. They are directly utilizing that. That is going to call as unprocessed and also primary biofuels. And coming to the secondary biofuels, these can be divided into as three. In first generation, here I am going to come with the your thematic subject of food security. So ethanol, butanol, and hydrogen are any other biofuel that are going to be produced from the sugar and starch feedstocks. Okay, so these directly affect the price and security of the food materials. Okay, so that's why previously in last decade, people have seen that the which for which you are going to give the importance, either for the fuel or for the food. But the what of our human beings is the first food. Why? Because without food we cannot live. So because of that, we have to have some alternative feedstocks, not direct products of the plant material, that is sugar and the starch materials, and the diesel which is going to be generating from the oil, that is not it is going to be not produced with the unprocessed or uncooked oils, that is come under the first generation biofuels. Coming to the second generation biofuels, that are ethanol and butanol and other biofuels produced from lignocellulosic biomass. So here the topic of my interest is coming to the matter. And then you are also having the third generation biofuels, hydrogen and methane, and also other butanol and ethanol, which are going to be produced through the algal biomass and the marine residues, which is going to be the third generation biofuels. So this is just I am giving you the introduction or background of the why, what is the importance of the biofuels, and why we need to utilize the whatever the agricultural residues to give the more food security to the human being. And these are the countries that are going to be producing the bioethanol from different types of the substrates. And this is also the pictorial representation. India and Thailand, Indonesia, and many other countries are there which are going to be producing from the different substrates. So Brazil, which is there here, it is going to be producing a very good amount of the ethanol from the sugar cane, direct sugar cane juice. And then coming to the substrate which we are utilizing, we know that first generation fuels are utilizing direct sugar or starch, and second one is the lignocelluloses, and third one is the marine biomass. And coming to these lignocelluloses, where from we are going to get the lignocelluloses? So all these lignocelluloses are come from the plant as a plant material. So we can get this from the all the crops, that is whatever agricultural crops, and also the forest waste, and also the grassland waste, and also industrial waste, all can be comes under this lignocellulosic waste material. We cannot say as a waste, we can residue, and this biomass, this we can convert into the valuable product. So this is the scenario of Indian crop and crop residue. And here we are going to be having the different types of the crops: sugarcane, groundnut, mustard, and also the cotton, wheat, uh, many paddy, which is going to be the major staple food in India, and then other thing is the wheat. And then what are the size of the crop residue that is going to be generating from these crops? So wheat is generating that is somewhat very high than other here, whatever here crop residues. And then other thing is the paddy, and next the banana crop waste or crop residue. And then we are having the sugarcane residue. Here you can see these residues which are going to be present. The sugarcane leaves, tops, and all going to be having, that is 7,787 kilotons per annum. It is going to be the surplus amount. And then coming to the sugarcane bagasse, which is a byproduct of the sugar industry, which is generating a high amount compared to the all the substrates that are available. And coming to the banana leaves, it is also going to be generating comparable size with the paddy straw, which is a very big portion that is going to be producing. And what people are doing with this, after the crop is completed or harvested, they are used to burn. And you might have seen in newspapers that the Delhi air pollution is going to be increasing due to the Punjab, the wheat and the paddy waste burning. So with that, because of that, it is going to be generating lots of whatever environmental pollution, and you are not utilizing that residue for the production of the valuable products. So we can utilize that to produce the valuable products like the biofuels, biochemicals, and other biological products which under the biorefinery concept. You might have known that the whatever the oil refinery, chemical refinery concept, from the crude oil you are generating some hundreds of the byproducts. First one is petrol, kerosene, diesel, and lactic acid, and ethanol, and butanol. Many are going to be extracting through the whatever the chemical refinery. And coming to this, whatever this biorefinery, where you are going to be utilizing the biological materials to produce n number of the products. So first the biomass is going to be convert into the simple sugars, and then they are going to be convert into the valuable products through the fermentation or any other process of the extractions, or any other process of the whatever the purification and separation processes. So here what you are going to, which is one of the very important way of it, biorefinery is, so whatever they generating the carbon dioxide and other greenhouse gases, especially the carbon dioxide generated from the biomass when you burn it or utilizing it, and that in turn utilized by the crops and grow fast. And once again, the same crops are going to be utilizing in the biorefinery to produce biofuel, to produce heat, to produce electricity, to produce biomaterials like paper and other things. And you can also utilize this ash and recycle them, and it is going to be the finally whatever you can prepare the some other materials which are going to be the end of the life of biomass. So because of this, the biorefinery concept is totally environmental friendly and also sustainable process. The sustainable is nothing but it has to be live for longer periods without any hurdles. That means the supply of raw material is very, very important. And then coming to this lignocellulosic biomass, where from you get this lignocellulosic biomass? Agriculture residues, forest residues, municipal solid waste, industrial waste, energy crops. So just I am little bit fast to complete my presentation. So within that, all these, the agricultural residue, forest residue, municipal waste, industrial waste, energy crops, all can be converted into the biofuel. But one hurdle is there, bottleneck of this, a big challenge to convert this cellulosic biomass into the biofuel. Why? Because this sugar and starch materials can directly utilized by the microorganisms and convert them into the respective bioproducts. But the cellulosic biomass cannot be utilized directly by most of the microorganisms that are still known and isolated and identified ones. What we need to do? First, we need to convert them into the simple fermentable sugars. And then you can convert into them, give as a feed to the microorganism and convert them into the a variety of the biofuels. So this is the structure of lignocellulose. The cellulose is going to be the present in the whatever wrapped by the two other molecules, that is lignin, another one is the hemicellulose. Finally it is there, the cellulose will be there, and this crystalline cellulose or amorphous cellulose is going to be the one which can be converted into the sugar, that is monosugar, that is glucose. The cellulose is a homopolymer which is going to be the beta 1,4 linked, uh, the glucose linear polymer, no other molecules will be present. And coming to the hemicellulose, it is a heteropolymer having the xylose, mannose, arabinose, galactose, and little amount of the glucose. And coming to the lignin, which is going to be the adhesive, and both of these, top of this, it is going to be protecting the attacking of the microorganisms of this cellulose. Because of that, cellulose is the backbone of the whatever the strength of the plant material. So because of that, the hemicellulose and lignin are going to be protecting that by wrapping it in it. Okay, coming to this lignin contains mainly the three different molecules, that is guaiacyl and syringyl and hydroxybenzaldehyde. Okay, so these three are the main components, and these three constituents of cellulose is backbone, and it has to be uh, whenever if you want to utilize the cellulose or convert this cellulose into the whatever glucose, you have to separate this hemicellulose and lignin first. Then you have the possibility for the whatever touching of this cellulose. So then the cellulose is not at all utilized by the microorganisms, and the microorganisms need to be have the enzymatic system, that is beta glucosidase, whatever the cellulase system, the three different types of the enzymes are present and very essential for the hydrolysis. So before this hydrolysis, you need to be separate or extract the lignin and hemicellulose. And for that purpose, you need to do or go undergo one another step as a big very important step, that is pre-treatment. So in pre-treatment you are going to be separate or extract or the whatever lignin and hemicellulose, you are going to get the amorphous or crystalline cellulose alone. Okay, so in pre-treatment you have different types of the pre-treatments. You have the physical, you have the chemical methods, you have the biological methods. In physical methods, we are going to be uncatalyzed steam explosion, liquid hot water, mechanical combination, and high energy radiation. These are comes under the physical, and chemical, which are going to be the catalyzed steam explosion, acid pretreatment, one of the very important and mainly utilizing in industrial biofuel production, and high energy radiation, organic solvents or organo solvents utilizing for the separation of these cellulose, hemicellulose and lignin, ionic liquids and alkaline pretreatments, and ammonia fiber bar the freeze explosion, which is going to call as apex, and biological treatment, treating with the lignocellulolytic microorganisms like basidiomycetic microorganisms or debryomyces, fungi, and some other microorganisms which having this cellulolytic system. And when this pre-treatment should be very efficient, it should be a cost effective, it should produce high sugar yield and increase the surface area to mass ratio to give the surface area to attack the enzyme system and get the good yield of the sugar, and maximize the enzymatic hydrolysis and fewer accumulation of inhibitory products. So very less amount of the inhibitory products also be generated. These are used to call as the plant materials, that is furfurols, hydroxyfurfurols, and polyphenols that are going to be present. So these three compounds are very inhibitory to the next step of the fermentation. That means these polyphenols and the furfurols and hydroxyfurfurols are going to be, HMFs are going to be inhibit the fermentation process. That's why whatever the ferment pre-treatment you are going to be selected need to produce a less amount of the inhibitory products and high amount of the sugar. Okay, so this is going to be the structure of the crystalline cellulose, no need. And coming to the cellulase system, that is the single enzyme cannot convert the total cellulose into the glucose. It needs the multi-complex system, that is endoglucanase, which is going to be cut. Endo means it is going to be cut wherever in linear polymer, it is going to cut inside of the polymer, and it need not to have any free uh, sites. Okay, either carboxylic or the amino residues. It can work on any place of the cellulose crystal. And then hexoglucanase, it needs this whatever the free ions, either carboxyl or amino system, and these are going to be generated, the endoglucanases are going to generate oligomers, which are also going to call as cellobiose. And then the second system is going to be generate the most of the times the glucose little amount and some other cellobiose, And then the final one, beta glucosidase, is going to be produced the monomer of the glucose from the all oligomers and also the cellular biohydrolases. So that's why whatever this microorganism, it should have these three. And this is the how the hydrolysis is going to be taking place. And then after hydrolysis, what you need to do? You are going to do the utilizing that sugar, you need to go for the fermentation. So there are three different types of fermentations are available industrially people are carrying out. One is batch fermentation, fed batch fermentation, and continuous fermentation. In batch, whatever the feedstock, whatever the sugar source, you are going to give at once and close the fermenter and wait until the fermentation incubation time is completed. Then coming to the fed batch, you are going to feed the fermenter intermittently or regularly. And then the continuous, you are going to pump the new media continuously and take out the whatever the fermented products continuously. Each and every method having its own advantages and disadvantages. And where I am working or our laboratory is working. Our laboratory is working isolation and characterization of solventogenic and also the lactic acid bacteria. Why? Because we are mainly focusing on biofuel and also the biochemicals and optimization studies of fermentation parameters, which are very important to get the good amount of the bioproducts. And then butanol and lactic acid production from the sugarcane field residue and butanol and lactic acid production from banana crop residue. And in addition to this, the butanol and also the lactic acid production from the industrial waste of the mango processing industry, where we are going to generate a huge amount of that is 30 to 40% of the waste from the mango industry. And then what are the methods or the steps that are going to be involving in our laboratory? People are doing research that mainly the media is going to be developed for the isolation and isolation of the solventogenic and lactic acid bacteria. And after preliminary screening, you go for the morphological and physiological identification and then molecular characterization and also the identification of a microorganism, and then preparation of agricultural residue, selection of the agricultural residues, and then prepare them into the simple fermentable sugars and go for the fermentation. So before the fermentation, we are going to characterize this agricultural residue before pretreatment, after pretreatment, before enzymatic hydrolysis, after enzymatic hydrolysis. And then we are also going to be analyze the alcohols, organic acids, polyphenols, furfurols through with the help of the HPLC, GC, and other systems. And then coming to the schematic, how we are going to go for the isolation. So for that, we will select any source for the sample first. Which sample you are going to be having in literature, go through the literature, and then you can select the sample and then go for the serial dilution. You might have known very well, and then go for the gram staining and the microscopic biochemical test, molecular characterization, and identification. So here for the whatever solventogenic bacteria, which are going to be the anaerobes. These we need a special system. That means the oxygen is toxic to these microorganisms. Because of that, you need the anaerobic glove box or anaerobic jars to cultivate them. In our laboratory, we procure this anaerobic glove box in DST project, and we are utilizing to isolate and also the production of the whatever biobutanol. And this is need to be utilized, not the simple whatever test tubes. Why? Because the oxygen is toxic to this microorganism, Clostridium, especially the solventogenic bacteria. Because of that, you need to utilize the serum vials. So in that also we can do this with the help of the serum vials. We can do the isolation by the two methods. One is hang gate method and another one is the stick plate method. If you want to do the stick plate method, you need compulsorily either anaerobic jar or anaerobic workstation. And we have isolated some 11 or 10 Clostridial solventogenic bacteria. From that, we have selected two which are going to be producing good amount of the solvent and identified them as Clostridium bigeninki and another one is Clostridium saccharoperbutyl acetonicum. So this having the a very good property that it can utilize a little quantity of the xylose, which is a component of hemicellulose. So that's why this organism is good. And then this is the same images of the microorganisms, Clostridium bigeninki and Clostridium saccharoperbutyl acetonicum. And coming to the molecular characterization of the lactic acid bacteria, we do the same and identified a lactic acid bacteria in our laboratory recently, that is Pediococcus pentosus from the idli batter. So idli is one of the staple breakfast in South India. That's why we have selected that for the isolation of the lactic acid bacteria. And we got the Pediococcus pentosus, which can grow a little amount of the pentoges, which is very important character, and it also have the character of it can grow above 45 degrees and produce the lactic acid at 40 degrees also. And this is the whatever same image of the Pediococcus pentosus. And then coming to the biofuel production solvent.
Interruption.
Why butanol? Why
Doctor.
butanol? Why butanol? Why?
Sorry, doctor. Sorry for the interruption. Actually, your presentation time is up. So maybe we can change to question and answer.
No, I can
Or
complete
if
within
anyone have any
five.
questions, you may present your question.
Okay, within 5 minutes I will complete it.
Hi, Doctor. My
Ah,
name is Dr. Raimi Muhammad Ridwan. I have a question to you.
ah.
About the processing of the biofuel. Is it more effective that we only use the chemical to process the cellulosic material for the biofuel? Or is it the combination of chemical and biological? Because I actually see that you focus on the isolation of bacteria for biological. So perhaps from my previous experience, I actually listen to the top people actually go with a chemical treatment. So do you combine chemical or biological, or do you just use either one? Thank you.
Uh, well, a very good question. That we are utilizing the chemical pretreatment, and then hydrolysis is going to be done with the help of the cellulase, and we are also isolated some of the aspergillus nidulans, aspergillus nimbus, some organisms which are going to be helpful in the pretreatment and hydrolysis of the whatever cellulose into the simple sugars. So this the work is progressing. We are utilizing both combination of chemical and biological.
Okay, thank you, doctor. Any other questions? Okay. We would like to thank Dr. L. Veeranjaneya Reddy for the speech just now. Now it's time for the third session of oral presentation. I will pass to the moderator for the session.
Thank you for the organizers for giving me this opportunity, and all the best for our successful completion.
It's for the next session, right? Or for the student session, right? Bismillahirrahmanirrahim. Alhamdulillah Rabbil Alamin wassalatu wassalamu ala ashrafil anbiya'i wal mursalin wa ala alihi wa sahbihi ajma'in. Subhanaka la ilma lana illa ma 'allamtana innaka antal alimul hakim. Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli. Thank you very much indeed to Mrs. Chairperson, and greetings to all, especially to the evaluators, Associate Professor Dr. Zul Arif, and also Dr. Laila, as well as other lecturers and officers of UMK inside the room. So we're going to have our third session on physical presentation. There will be 4 presenters are going to do the presentation today, this afternoon. And perhaps before we start our seminar, if I may remind the presenters that your time is actually 15 minutes for each presentation, and followed by question and answer session. So without further ado, I would like to call upon and invite Ms. Chan Ke Xin to present her work on synergetic effects of bitter gourd honey with treated Cornsilk extract on total phenolic content. The floor is yours.
Thank you for Prof. Lokman for the invitations. Slide. Okay, good afternoon for all for our moderators, Prof. Lokman, and others, the 2 panels, and also others audience. Thanks for coming in. Today I would like to present my result on the total phenolic contents of bitter honey with the treated Cornsilk. So first, I would like to have a brief introduction of bitter honey. It's just similar as the normal honey, which is a natural sweetener and is used in the traditional therapy, as we know. And of course, this is why it's called a bitter gourd honey, because bitter gourd honey is like the bees only collect the honey from the bitter gourd flower. So it's a monoflora honey, yes. So for Cornsilk, actually it's the purple yellowish stigma from the corns, and the part that I take is the baby Cornsilk. So I take the Cornsilk from the baby part, which is also one of the byproduct from the corn industry. So total phenolic content is to test the phenolic compounds that contain in the product that what I test, yes, just in short view. So for the methodologies, for the treatment part, I will do the blanching treatments, or normally we call heat treatment as well. So for the fresh Cornsilk, there will have 3 treatments, which is the non-treated one, and then we'll undergo steam blanching and hot water blanching. For each is 2 minutes separately. And for the drying process, I will dry in 55 degrees Celsius, and there is 2 drying temperature period, which is 24 hours or 36 hours. So for the extractions, I'm using the homogenized extraction methods, with the samples will mix with the distilled waters and then homogenize under 1000 RPM for 50 minutes. And after filter it, I will centrifuge it. And then I will store it in a negative 20 degrees Celsius for further testing. So to preparation, the Cornsilk and honey extract samples combinations is follow the ratios as below. So, and for the testing, so total phenolic content is using the common Foley concoctors reagents, and then stand for 6 minutes, and then I will add the sodium bicarbonate to stop the reactions, and then store in the dark condition to let it stand more well for 130 minutes, and I will observe the absorbance values and the 765 nanometers. So this is a result for the total phenolic content of the treated Cornsilk extract that dry for 24 hours and mixed with the bitter gourd honey with different ratio. So as we know, the blanching treatments is have a slightly increase on the total phenolic contents in the Cornsilk, because sometimes the heat treatments may burst the cells and let it let the compound easily to be extracted during the extraction process. So here is for the 36 hour, which is more drying, longer time, but compare, maybe it's difficult for you to compare, but compared to the 24 hours and 36 hours, for steam blanching and non-treated blanching, non-treated one, the total phenol content is a bit lower when we increase the drying time. Yes. So because this is a short view, I just want to see whether there are any reactions that heat treatments will react with the Cornsilk, which will preserve the Cornsilk phenolic content, whether the honey is helped or not. But according to the result, that is not very good for me because I do the statistical analyticals. There's not much difference in between them. So for my formulations. But of course, the potentials for the bitter gourd honey, whether it can be enhance the Cornsilk. The potential by using bitter gourd honey to enhance the Cornsilk of polyphenol contents can be discussed as well. So I showed this is my own result because I tested it, which is the formulation is not what I want. Yeah, the result doesn't have much difference. So I didn't. So we will go for the new formulations in my new. This is the previous one. That's why I only show for total phenolic content. Yes. Is there any questions?
Right, thank you very much, Chan. So I open the floor for any question from the panel first.
Okay, thank you, Chan, for your very short presentation.
Yeah, just a short and sweet.
Okay, yeah, I understood from your presentation, it looks like you just present a part of your experiment, yeah?
Yes, it's part of my experiment.
So it could be nice if you actually inform what is the significance to do this research, why you are doing this, what is the importance of the bitter gourd honey.
First thing first, why I chose the bitter gourd honey is because bitter gourd honey is currently new in the research world. Even though I searched for the paper to want to understanding the bitter gourd honey is very limitations. So for us and in Malaysia, there are honeybee, there's bee farming, they are producing bitter gourd honey. So this is what make us want to choose bitter gourd honey for testing on it. And secondly, is why I want to choose Cornsilk extract, because Cornsilk is part of the byproduct in the corn industry. So I'm curious on whether can I use some like little bitter gourd honey to enhance the phenolic contents in the Cornsilk extract or not. I want to see their synergetic effect in between them.
So is there any therapeutic effect at the end of your experiment? Means this is just your result only, but what is the output later?
The output later is just actually is like thinking like having a beverage or like have more choices for the market, which we can get a lower, which is cheaper antioxidants or anti-diabetics raw material compared on honey itself. Is want to compare in terms of the expensive honey and use less honey, but which can enhance some byproduct, which can have a similar similar value to give to the
So in
future.
that case, you have to your cost analysis also, I think.
Yeah. There's others analysis because like this is just a part of like because formulations is a trial and errors. Yeah.
Okay, thank you.
Thank you, Dr. Laila. And you have no question? I have few questions, if I
Sure.
may. Number one is, how do you make sure that it is a bitter gourd honey? Do you produce this honey or you bought it?
Okay, first I bought it from the bee farm, which is their labeled as bitter gourd honey. But if
Okay,
you want
now
to test
how
on
they,
it,
they produce this bitter gourd honey instead of other kind of flowers? So they conceal the bees just to get the honey from that tree only.
So it's common route for because that time when I purchase the honey, I will write to request to visit their farm directly. But due to that time it's MCO, so they are say strictly forbidden for us to enter. But they describe me is like they're having a big bitter gourd farm, so they have the honeybee inside in that area, so they have provided sufficient flower for them to go. But if we want to go to truly to test whether it is a bitter gourd honey, we can go to the microscopy to test for the flora. We have to collect the pollens inside it and observe whether the pollens is whether
So the farm is not enclosed with netting or something.
they didn't discuss much of that because I cannot visit them, so I
Okay.
don't know whether they like throwing through or not.
All right.
Yeah.
Question number 2 is on your analysis.
Okay.
You are using UVVs, right?
Yes.
765 nanometers.
Yes.
In reference to what molecule?
Oh,
In-
but
In reference to what molecule for 765 nanometers?
is to test for the weather. That's the name of it. Sorry. So it's to test for the intensity of the molybdenum tungsten blue. The one of the indicator will change the blue color. Yeah.
That is the indicator, yes.
Yes.
But
I want to,
the actual phenolic molecule that you are investigating is what actually at 765 nanometers?
sorry about that because it's a total phenolic compounds. I didn't specify on that point.
Okay.
I just choose this wavelength is just it's
Is-
from
is that
few
one-
reference for that. Yeah.
Is it one molecule specifically or a group of molecules?
A group of molecule is a group of the phenolic compounds group. Yeah.
Okay, perhaps in next time that you can list out all those molecules and people can know that what is actually those phenolic molecules.
Sure, thank you.
And instead of picking up at the end of the process, because you are showing your data on the end of the process, right?
Yes.
Can I see the data, the tables of your results?
Is one of the table.
Yes. So these are extraction conducted in 24 hours, right?
Drying in 24 for the conceal after blanching.
Okay. So concept. So you are getting the concept TPC, right?
Yes.
The phenolic compound from the conceal
And also the mixture with honey,
with honey.
based on the ratio.
Do you have data? I suppose you have data between honey and conceal. So which one is actually higher in TPC?
Higher in TPC is the honey. You see the lowest bottom one, 0/5, the ratio.
Oh, okay.
Yeah, that one is a honey. Of course,
Then
honey it will be stronger because honey is we already know honey have.
So you are synergistically you expecting to have more. So the 50 is 55. So combination should be higher, because because you have 55 for Cornsilk and then you have 64 for honey.
Yeah, actually
So-
I do more than that, but just this is summarized, which is more easier to like, I pick it up in the middle. So it's easy to compare.
So the unit of this is milligram, right?
Yeah.
Perhaps if you can report for better understanding
Okay, sorry
In the-
about that.
in the form of percentage or parts per percentage from your raw material, percentage of GAE, milligram of GAE per gram of your raw material. That would be very nice.
Thank you. Thank you for your.
Any other question? Okay, I allow question from other members of the floor. Soalan? Ya, takutnya. Takut nanti you all present dia pula tanya. All right, thank you very much, Chan. A very beautiful presentation indeed. Thank you.
Thank you very much for everyone.
All right, we go on with the next presenters. Oh, nak kena isi sekarang ya? Okay, I will do it. Yeah. Okay, please allow us to fill up the form, and then we start with Rival. Okay, our next presenter, PD05 Rival Yuhendri from Indonesia on microscopic assessment of leaf, root, and rhizome of Curcuma Sumatrana.
Oke, terima kasih, Profesor Lukman. Sebelumnya ini sangat tegang sekali, saya rasa dari pagi. Ya, apa namanya? Sangat menegangkan. Ini mungkin ini pertama kali bagi saya untuk simposium ini, menurut saya apa ya? Karena saya dari Indonesia. Ya, sebelum saya mulai izin Prof, karena saya bahasa Inggrisnya tidak begitu bagus ya, mungkin I speak Indonesia mungkin insya Allah nanti. Oke, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Oke, sebelumnya saya mengucapkan terima kasih kepada panel sekaligus hadirin yang datang pada siang menjelang sore hari ini. Oke, mungkin lanjut saja. Perkenalkan terlebih dulu, nama saya Rival Yuhendri. Saya dari Padang, Indonesia. Mungkin dari sini agak jauh ke sini ya, tapi insya Allah lah untuk belajar ya. Oke, pada kesempatan ini saya akan coba berbincang tentang ya microscopic assessment of leaf, root, and rhizome of Curcuma sumatrana. Ya, di sini saya akan coba. Oke, sedikit kita berbicara tentang introduction ya, sampel di mana sampel yang saya jadikan sebagai objek research saya ini adalah Curcuma sumatrana ya. Mungkin barangkali kita tidak dari segi bentuk secara morfologi ini hampir sama dengan Curcuma longa. Ya mungkin kalau kita untuk memasak Curcuma longa yang sering kita gunakan kunyit, mungkin ini bentuknya mirip sekali dengan Curcuma longa ya. Secara taksonomi pun mereka memiliki kekerabatan yang sangat dekat, yaitu sama-sama genusnya adalah Curcuma. Itu yang pertama yang ingin saya sampaikan. Oke, selanjutnya bahwasanya Curcuma sumatrana kalau kami dari Indonesia itu menyebutnya adalah kamuniang, nama daerahnya. Kemudian ini merupakan tumbuhan endemik Sumatera Barat, ya khususnya daerah Padang. Kalau saya berbicara ini mungkin ya nanti khususnya di daerah Padang. Kemudian berdasarkan IUCN ini masuk ke dalam kriteria endangered species ya karena kelangkaannya itu sendiri. Itu berdasarkan IUCN 2001. Kemudian secara morfologi seperti yang saya sampaikan sebelumnya bahwasanya Curcuma sumatrana itu memiliki morfologi yang mirip dengan Curcuma longa ya atau Curcuma genus ya seperti dari kita lihat dari bentuk leaf, flowers, rhizome and root-nya itu mirip. Namun di sini yang agak membedakan secara morfologi barangkali ini bentuk rhizome-nya ini dia ini lebih mirip dengan lengkuas, mungkin bahasa kitanya sayanya itu lengkuas akan mirip dari itu. Secara rhizome ini agak sulit kita mendapatkan. Oke, secara taksonomi seperti saya sampaikan sebelumnya bahwasanya dia masuk ginger untuk klasifikasinya, genusnya adalah Curcuma, spesiesnya adalah Curcuma longa. Sedangkan secara distribusi yang saya sampaikan sebelumnya bahwasanya dia endemik Sumatera Barat seperti mungkin seperti gambar di samping. Oke, next untuk problem of study bahwasanya Curcuma sumatrana ini adalah endemik Sumatera Barat dan masuk ke dalam kriteria endangered species itu berdasarkan IUCN 2021. Kemudian sejauh ini belum ada kajian research tentang karakteristik anatomi ya. Sejauh ini karena spesies ini rediscovery untuk statusnya adalah rediscovery 2021 sebenarnya untuk spesies itu sendiri sudah pernah ditemukan Microscopic 1861 secara historinya mungkin sedikit saya bicarakan itu. Kemudian secara objective study ya, tentu di sini kita akan melihat seperti apa karakteristik anatomi ya, baik itu dari leaf, rhizome and root dari Curcuma sumatrana ini. Karena untuk referensi itu sendiri ya, setelah saya konfirmasi kepada LIPI, belum ada secara baik itu kajian anatomi maupun kajian phytochemical sejauh ini belum ada kajian, hanya sebatas dengan kajian tentang taksonomi itu sendiri. Oke, next material ya, di mana sampel ini memang saya ambil dari Indonesia, kemudian saya identifikasi dari Universitas Andalas dibantu oleh Dr. Nurenas, kemudian sampel tersebut saya simpan di herbarium Anda. Selanjutnya untuk metode, di sini kita menggunakan metode SAS ya. Ini mungkin metode yang sangat lama sekali kita gunakan kalau kita membuat suatu preparat permanen anatomik, ini yang sering kita gunakan metode SAS. Ya, parafin method ini yang sering kita gunakan, di mana untuk prosedurnya itu sendiri, saya rasa ini cukup panjang untuk prosedurnya sendiri, di mana sampel yang kita koleksi tersebut kemudian kita lakukan dengan fiksasi, ya. Kemudian kita lanjutkan dengan aspirasi, kemudian kita lanjutkan dengan dehidrasi, di mana untuk timernya ini cukup bervariasi. Karena saya rasa untuk membuat suatu preparat permanen ya, mungkin secara metodologinya itu hampir sama. Kalau kita membuat preparat permanen tumbuhan dan hewan itu hampir sama secara proses untuk pembuatannya itu sendiri. Kemudian dilanjutkan dengan infiltrasi, kemudian embedding, mounting, sectioning, mounting, kemudian staining, kemudian mounting lagi, kemudian observation. Setelah kita dapatkan adalah kita telah menghasilkan suatu preparat permanen, baru nanti kita observasi di bawah mikroskop. Selanjutnya adalah result and discussion ya. Berdasarkan hasil yang telah kita dapat secara mikroskopiknya, kita dapat secara preparat permanennya, kemudian kita amati di bawah mikroskop, kemudian kita lihat parameter apa saja yang mungkin kita lihat di sana ya. Di sini dari 3 organ yang kita amati, leaf, root, and rhizome, ini kita lihat diameternya, leafnya itu seberapa size-nya dalam bentuk dalam mikron. Kemudian di sana adalah seperti diameter leaf, kemudian rasio stomatanya. Mungkin nanti akan saya lihatkan juga gambarnya seperti apa ya. Di sana beberapa parameter yang saya perhatikan seperti diameter leaf, rasio stomata, kerapatan stomata, kemudian diameter kristal atau perkiraannya seperti itu, kemudian diameter epidermis, diameter atas dan bawah, kemudian diameter mesofil, tinggi trikoma, kemudian luas pembuluhnya di sana, kemudian untuk rootnya itu sendiri saya coba amati seberapa diameter root itu sendiri, kemudian diameter cortex, kemudian diameter endodermis ya, diameter xilem, diameter empulur. Kemudian pada rhizome kita temukan adanya pati. Ini mungkin lazim kita temukan pada rhizome dari curcuma ini, sangat banyak sekali pati terdapat di sana. Kemudian juga kita menemukan kristal ya. Kemudian di sini juga kita melihat korteksnya, kemudian kita juga amati endodermisnya, diameter endodermisnya itu sendiri. Oke, seperti ini kira-kira yang kita peroleh dari preparat permanen, kemudian kita amati di bawah mikroskop itu dari daunnya itu sendiri, ya sayatan pada dermal dan paradermal atau secara transversal ataupun longitudinalnya ini. Kemudian ini adalah rhizome-nya. Selanjutnya ini adalah root-nya ya. Oke, constellation. Beberapa referensi yang saya coba bahwasanya setelah saya amati struktur anatomi dari daun, leaf, rhizome, and rootnya, kemudian saya coba compare dengan spesies lainnya, Curcuma longa atau Curcuma zorodia. Secara keseluruhan itu memiliki struktur yang hampir sama saja ya. Secara keseluruhan dia mirip saja, baik itu secara struktur. Yang membedakannya di sana ketebalan atau diameter dari jaringan itu sendiri ada yang tebal, kemudian sel penyusun jaringan itu sendiri ada yang 1 sel ataupun ada yang 2 sel. Jadi secara keseluruhan itu memiliki struktur yang sama. Kemudian yang lebih spesifik di sana yang pada daun yang saya coba amati, bahwasanya tidak semua jenis daun curcuma itu ada kita menemukan kristal ya, kristal salat mungkin barangkali yang dapat kita identifikasi pada daun itu sendiri. Mungkin secara untuk constellation-nya saya rasa untuk hipotesis kita memiliki struktur yang sama secara anatomi, baik itu daun, leaf, root, and rhizome-nya itu sama. Selanjutnya ini adalah referensi yang bisa kita gunakan. Oke, terima kasih.
Alright, terima kasih Rival. Thank you very much. Any question from the panelists?
Ya, Pak Rival, saya ingin tahu apa manfaat tanaman ni, dan kalau bisa Bapak tekankan problem statementnya lebih luas lagi lah, agak very mild kan problem statementnya. Jadi mau tahulah apakah adakah diguna secara luas di Sumatera Barat atau macam mana?
Oke, terima kasih Prof. Untuk pemanfaatan di sini beberapa masyarakat khususnya daerah Maninjau, Bukit Tinggi, mungkin yang daerah sana, masyarakat setempat memanfaatkan daunnya itu, air rebusannya itu digunakan sebagai obat gatal-gatal barangkali, untuk obat gatal-gatal. Kemudian mereka juga menggunakan sebagai, karena daunnya mungkin sebagai bungkusan, saya tidak begitu pasti seperti apa metode penggunaannya dari masyarakat setempat, karena beberapa referensi, karena saya coba menjalani daerah Sumatera Barat itu, memang secara keseluruhan masyarakat itu tidak mengenal jenis Curcuma sumatrana ini hanya pada daerah-daerah tertentu saja mereka menggunakan. Mungkin dari situ untuk pemanfaatannya.
Dr. Laila? No, you don't have question, no? Very kind of you. You are mentioning that Bapak ada mensikan tentang endangered species ya.
Iya.
Kenapa dia jadi endangered itu?
Gini, berdasarkan kebetulan untuk spesies Curcuma sumatrana saya koordinasi dengan LIPI itu atau sekarang itu namanya Riset BRIN dari peneliti dari Indonesia itu sendiri, bahwasanya kebetulan untuk rediscovery rediscovery spesies Curcuma sumatra ini adalah Dr. Adriani dari LIPI. Kemudian saya terus komunikasi dengan beliau, bahwasanya beliau menceritakan kepada saya untuk proses penemuannya sendiri, untuk penemuan rediscovery-nya sendiri, memang Dr. Adriani itu ke Padang untuk mengambil sampelnya untuk diidentifikasi. Kemudian mereka itu menceritakan untuk distribusi dari keberadaan Curcuma sumatra ini memang betul sudah terancam punah, karena dari segi pemanfaatan itu sendiri masyarakat tidak membudidayakannya karena mereka menganggap bahwasanya Curcuma sumatra ini seperti tumbuhan liar yang hanya tumbuh di ladang-ladang mereka, kemudian karena mereka tidak memanfaatkannya, mereka secara ini untuk memusnahkannya gitu, seperti itu untuk ini kriteria. Tapi sepanjang kemarin yang ingin saya ceritakan untuk kriteria IUCN ini, dia menceritakan bahwasanya sedang diajukan karena sudah masuk zona merahnya red ini, tapi sampai sekarang saya belum dapat informasi apakah sudah diterbitkan Curcuma sumatra ini masuk kepada IUCN ini. Seperti itu, Prof.
Oh, jadi apa yang saya faham ialah endangered ni maksudnya ada penyakit disebabkan oleh penyakit ataupun dimusnahkan oleh manusia ataupun dia punya habitatnya musnah. So mungkin Bapak boleh terangkan dari sudut itulah nanti dalam tesis ya.
Okay.
Soalan kedua saya adalah dari segi scope of research lah. Adakah ini akhirnya akan dijadikan makanan ataupun kosmetik?
Ya, kemudian ini adalah mungkin bagian dari part riset saya. Setelah saya mengkaji secara anatomi, kemudian sekarang kita try untuk phytochemicalnya. Setelah nanti mungkin kita dapatkan per compound ataupun compound tertentu yang bisa kita manfaatkan ya sebagai obat atau sebagai lainnya. Mungkin setelah itu mungkin kita bisa berbicara tentang apa manfaatnya secara realnya.
Bukan dalam you punya PhD punya scope nah.
Yeah.
So you punya awak punya PhD punya scope hanyalah study on the characterization of the plants, right? Begitu.
Tak, di sini kita anatomy and phytochemical analysis.
Oh, so there will be a chemical analysis, and then you have certain analysis using GCMS.
Yes, insyaAllah.
That's great. Okay. So terima kasih banyak Bapak Rival. Thank you very much indeed. And please allow any question from the floor.
Iya, dokter. Phytochemical. Ya kebetulan dokter saya baru jumpa tadi saya nanya-nanya yang mana nih yang dokter salah satu panel saya ini saya tanya-tanya. Kemudian gitu ya dokter, terima kasih. Baru jumpa sekarang.
Ini experts in herbs.
Iya.
So
Indonesia. Iya. Sekarang sedang ini dokter, sedang phytochemical, sedang ekstraksi. Sedang mengolom, sedang jalan kolom insyaallah lah. Iya, di PG Lab. Iya, thank you. Ya, thank you.
Alright, terima kasih Bapak apa nama?
Rival.
Jarang-jarang kita sebut nama ini di Malaysia.
Oke, terima kasih.
Terima kasih. Okay, can we allow us big applause to Bapak Rival? Okay, tak sabar ya Cik Firas ya. Next presenter will be Muhammad Firas Hamizan bin Hassan, who will be presenting on optimization of grass jelly formulation by addition of gelatin, physicochemical and textural analysis. The floor is yours, Firas.
Test. Assalamualaikum and a very good evening to Prof. Luqman, Dr. Laila, Dr. Zul and Dr. Shamsul, and to all the audience in this ceremony. So I further my presentation today. So my name is Muhammad Firas Hamizan bin Hassan. I will be presenting my research on optimization of black grass jelly formulation to increase floating rate and reducing leaching. So this is my presentation outline. So Mesonogenesis L exerts many physiological effects. It is essential to maintain the physical appearances of BGG, black grass jelly, until it's reached the consumer. This is why at first the research is this is why I conduct this research at first. So food and beverage quality decide customer acceptability. The leaching of the color cause the quality of the black grass jelly commercial drink to decrease. How I overcome this situation is by implementing carrageenan and gelatin as alternate binder for the formulation. My null hypothesis is the application of carrageenan and gelatin does not affect the physiochemical characteristic of BGG or prevent leaching, while my alternate hypothesis is the application of carrageenan and gelatin affect the physiochemical characteristic of BGG or prevent leaching. This is my objective. My first objective is to determine the effect of the selected gelling agent on the texture and color of black grass jelly, to determine the leaching rate of black grass jelly prepared using selected gelling agent, and lastly, to determine the sensory scores of optimized black grass jelly. This is my gap study. So an overview of literature review. Susmita and Ling have summarized that bioactivities of this herb, antioxidative, anti-inflammatory, antihypertensive, and immunomodulatory agent. There are a few factors influencing the rate of leaching. It's the internal chemical reaction, physical response, and external stress from the surrounding environment. The implementation of a solidification agent is one of the crucial factors affecting the gelling quality during processing. Combined use with other gum allows for a broader spectrum of jelly textures and mouthfeel in these jellies. That's why carrageenan is well known for its excellent clean flavor release. The food industry, for example, manufactures gourmet dessert using gelatin for texture, foaming, and clarity, as well as stabilizing the food structures. So methodology part. This is my novelty of the experiment. A new optimized formulation of black grass jelly will be developed. Correlation in between selected gelling agent, physicochemical texture and consumer acceptance will be established for future studies. So I will start my research with preparation and development of a different formulation of gelatin and carrageenan by using design expert 13, sample preparation. And next, the color, pH, texture and floating rate of grass jelly will be analyzed. Leaching rate analysis will be evaluated using standard method. Optimizing the formulation will be analyzed and sensory score. Next, in the last part of the experiment, the data will be tabulated. So this is type of my sample and formulation. So for today's presentation, I will be focusing on the design expert part. So this is my analysis outcome. For today, I will be focusing on the leaching rate and floating rate. For the leaching rate, because the color exit from the jelly is yellowish, so two things, floating rate and color determination from B, yellowness. This is scatter plot analysis. So the blue color indicate that the relationship is moving toward negative, which mean if we, for example, we focus on pH value, if we add more carrageenan, the pH will increase. If we add more gelatin, the pH will decrease. So this is my optimization part. The thing is that if we refer to the analysis from design expert, we notice that there is no detailed correlation in between carrageenan and gelatin for floating rate, because if we plot the graph, it's actually a trend where only half of carrageenan and half of gelatin will give out the longer floating time. So if we put a little more carrageenan or a little more gelatin, so kalau macam we put more gelatin, the jelly will become, the texture will become so a little bit hard and the floating rate will decrease. While if we add more carrageenan, the texture of the jelly will become lembek sangat. So because we test the floating rate in brick 11 sugar, so the solution tend to penetrate the jelly and lower the floating rate for the black grass jelly. So the conclusion for today's presentation is that the correlation of gelatin and carrageenan toward BJG is studied. The rate of leaching are lower compared to traditional made black grass jelly. Thank you.
All right, thank you very much, Faris. Any question from the panelists?
Okay, thank you, Faris, for your nice presentation. You are doing the actually optimization, yeah? So grass jelly is already used for the breweries, yeah? Is it using for breweries, grass jelly? What kind of implementation for this grass jelly? People consume it, yeah?
Yes. Usually it's made for RTD, ready to drink punya minuman.
Yeah, breweries.
Cincau.
Okay. So when you want to remove the leaching problem that make the discoloration, all these things. So and then you have done already that you have shown
Yes.
here. So I think the presentation is quite complicated for your data. It's not clear. Besides that, when you add this gelatin and another one is what?
Carrageenan. Carrageenan.
Carrageenan. So how about the nutrition? Is there any effect of the nutrition in this jelly brevis?
For this, for my study, I will not touch the nutritional effect, but for as we know, carrageenan also act as anti-stress. So in term of nutritional punya value, mungkin boleh tambah untuk next study atau limitation for these studies.
But you are optimizing, okay? So I think it would be nice that you do yourself that how the effect of this. Okay, that's my advice. Okay.
Okay, I have several questions. Number one is optimizing the parameters. So identification of what parameters to be optimized is very important. And then you want to ensure that at the end, your product is of highest quality. So in terms of color, because you are using the color meter, so what would be the specific parameter that you are looking into?
Specific parameter.
Okay, apa parameter yang diukur tu yang merah-merah, biru-biru tu, putih tu? I mean the color of your jelly. So the blackness actually represent highest quality of your product.
Betul.
Kalau dia cerah, tak elok. Kalau dia gelap, elok.
Elok, betul.
So
cumanya yang untuk saya masukkan dengan leaching tu adalah bila kita test dengan
yeah, that will be my next question.
Okay.
So the color in here, the best parameter, the best quality of that darkness is in blue or red? Lagi gelap. Okay. So it is difficult to comprehend this very complicated Panama tables like this. So if I may suggest that if you want to explain things, you need to segregate into different effects. Leaching tempat lain, the color parameters analysis tempat lain, and then the texture also different. Kalau you taruh ini dalam you punya thesis, nanti you punya examiner akan marah ya. Nanti akan bagi markah yang rendah because you unable to explain to examiners in a sequence manner. Ya, dia tak boleh. Memanglah you sukalah, student suka letakkan everything dalam satu page, and then you pun mulalah explain based on your shot sendiri, and then examiners tak suka. So the principle is that you always try to present in a manner that people will be happy with your presentation. So perhaps our suggestion is that you segregate now. Segregate, okay? You clearly nak tunjuk effect apa? Effect of leaching rate tu you need to explain meticulously how do you measure the leaching. And then you study on the parameter of color, then parameter of texture. So you actually have different 3 subchapters on results and discussion on these three, and you must have at least three objectives lah. You understand what I'm saying? Back to your leaching rate. Can you explain a bit to us? How do you measure the leaching rate?
Okay, so measure my leaching rate using 2 machine, which is colorimeter and spectrophotometer. So for the calorimeter, I put the jelly into bricks 11 water solution with sugar. So I measure for 10-minute time interval, start with 10, 20, 30 until 90
So,
minutes.
Do you understand the term rate here? RAT, rate.
Perubahan
Your
yang
parameter, which is
color yang exert daripada jelly tu.
darkness. Perubahan the darkness over the time. Perubahan the darkness, kehitaman, kegelapan you punya solution over the time.
Tapi yang dekat sini yang kita ukur tu yellowness doktor, sebab saya ada gambar untuk tunjuk, sebab color air yang
It's okay. It's okay. You can select whether it's the redness ke, the blueness ke, yellowness ke, but you have to mention lah that you are measuring the intensity of yellowness.
Okay.
Okay, and over the time. And the most important data is actually the graph over the time, intensity versus time.
Terima kasih untuk jangan doktor. Cumanya untuk discussion hari ini saya
You don't have. You thought that it's not important lah to show.
bukan doktor. Cuma untuk discussion hari ini saya nak try saya punya ability untuk discuss design expert punya result. Saya ada, kalau in writing part saya ada letak daripada flow macam Dr. Lokman cakap. Cuma untuk presentation hari ini saya nak tengok ability saya untuk discuss design expert punya part. Sebab for this table, the only thing yang table ni nak tunjuk adalah correlation. Tapi macam saya cakap tadi, terima kasih sebab saya akan go through balik saya punya writing. Cumanya untuk yang ni lah. So macam table yang ni untuk kita lihat correlation antara carrageenan dan gelatin.
Okay. And when you use the word correlation, what is the most important parameter of correlation?
Important parameter.
The R square actually. So you have to plot the R square between carrageenan and that gel, and then you can calculate the R square. Then you can claim that there is a strong correlation between the two. That's so. All right, I suppose thank you very much indeed. And then there is a very good research actually. Kami tanya ni sebab nak bagi strength.
Tak. Okey. Saya untuk presentation ni sebab saya nak tengok ability nak present design expert punya calculation. Kalau macam yang nak cari relationship R square tu, memang design expert dia ada bagi siap lah. Cuma saya tak include the whole sebab tujuan objektif presentation hari ni adalah untuk tunjuk correlation tu sendiri.
Understand. Okay. Thank you very much indeed. Okay. A big applause to Faris, please. Thank you. So the next presenter will be Nurul Atira binti Muhammad Zuki on pre-treated black soldier fly larvae as sustainable alternative protein source for broiler chicken.
Assalamualaikum dan selamat petang. My name is Nurul Atira binti Muhammad Zuki alias Rosli. Today I would like to present my small part of my research, pre-treated black soldier fly larvae as sustainable alternative protein source for broiler chicken. And today the presentation outline, first we will go to introduction and then methodology and result and discussion and also the conclusion. Okay, for the introduction, according to the FAO, the world population between 2009 and 2050, that was expected to increase 70% in 2050. And this phenomena will create the high demand of meat, especially for poultry and fish. And the potential of insect protein in poultry diet has attracted much extension as a long-term solution for the animal feed production. And also insect at all life stage are rich sources of animal protein. And for my research, I use black soldier fly larvae. It has a high protein level up to 50%, as it can replace soybean and fish meal. For the information, broiler chicken digested less than 50% of BSF protein. The introduction of protease in this poultry diet had maximized the digestion by performing hydrolysis and breakdown. Protease treatment proved that BSFL protein could partially be hydrolyzed into small peptide. And the hybrid treatment, which is the combination of salt and protease, is good to treat BSFL, as it can denature BSFL protein and assist in refolding the enzyme. And for this study, there is 4 group of my research. For group, and each group I allocate 30 of broiler chicken, and for the first group we is control untreated BSFL, T1 50% of salt treated, T2 50% of protease treated, and T3 15% of hybrid treatment. And the objective, so today I just show the part of my study. First, to synthesize the salt-treated BSFL, protease-treated BSFL, and hybrid-treated BSFL meal. And then to correlate the influence of the treated BSFL on the meat growth, on the growth performance of broiler chicken. And today I will present the ADFI, average daily feed intake, average daily gain, ADG, feed conversion ratio, FCR, and mortality rate. Okay, and for the methodology, so first for the feed formulation and the feeding trial. Okay, before the feed formulation, at first I carry out the pre-treatment on BSFL, which is pre-treatment using salt, protease, and hybrid treatment combination of salt and protease. And then the mixture will be in the room temperature. In the room temperature for 24 hours, and then it dry at 45 degree, and after that I will grind to produce the powder, and after that I formulate the feed formulation and pelletize. And after that, for feeding trial, I give the feed to the chicken at libitum, which is lebih macam tu, for 35 days. And then throughout the feeding trial, vaccination program had been done, and the fecal collection and also the blood sampling. And so for the result, this is the result of ADFI and average daily feed intake. As you can see, the T1 starting from the week 2 until the week 5, there is highest feed intake. But you can see that the average daily gain, the weight of broiler chicken, T3 hybrid treatment show the highest weight starting from week 3 week 4 and week 5. And for the feed conversion ratio, in starter stage, we can see that week 1 and week 2, T1 and T3 show a low range of SCR, which is 1.05 to 1.29. Meanwhile, at week 3 and week 4, T3 and T4 demonstrated 1.76 and 1.88 of SCR value. But at the end of the week, T1, T2, T3 present the low FCR value compared to control group. So for the mortality rate, the highest mortality rate is in the control group, and the lowest mortality rate in T2 group, which is protease treated group. And for the conclusion, so we can conclude that the growth performance include average ADFI, ADG, and FCR. Also, the mortality rate were affected by the diet. And the FCR group T3 show significant different lah, slightly different between 4 group. And the mortality rate greatly affected in group, control group and T1 group. And despite of this outcome, the study showed that protease treatment and hybrid treatment group might be practiced in future to produce boiler feed, maybe in industry. That's all. Thank you.
Right, thank you very much, Nurul Athira. I can open the floor for any question from the panelists.
So what is the effect of consuming the lava? Well, I want to know whether this lava, is it a mixture with the early stages when you're doing the feed?
Yeah. From the day 1, day 0.
So the consumption of the broiler, is it morning time they are consuming this and evening time? Because normally poultry takes 2 sessions of consuming, right? Day, morning and evening. Right?
Yeah.
So they are taking both time.
Yeah.
So how's the impact of it? Meaning is it different from the outsiders are doing, the publics, I mean the other farmers are doing?
Different
in terms of of the size, the whatever, the health of the broiler.
Actually, I want to compare the control group, the untreated and the treated. And for the weight, we can see that at the end of this experiment, the untreated group show the very lowest in weight and also the highest in mortality rate. So for this experiment, I want to compare untreated and the treated only, not compared to outsider.
Okay, because Athira is actually the last person to present, so we have plenty of time to ask her a question. Why? It's okay because closing ceremony is 4:40, so we have another 40 minutes to grill her. You don't want to ask? Okay. My concern is your control group.
Yeah.
Control group you guna apa makanannya?
Untreated BSFL, dia formula dia sama je. Yang bezanya dalam tu
Still got BSF.
Ya, tapi
So
untreated.
You don't have a group that use common feed?
Commercial. Actually ada, tapi saya just hari ni saya nak bagi saya just nak compare between untreated and the treated.
Ya ke? Sebab maknanya kalau yang makan yang komersial tu rasanya dia apa lagi sihat dia.
Ya, betul.
Oh
Saya
my
pun tak
goodness.
tahulah formula apa yang ada dalam tu. Maybe dia letak booster sebab saya tak ada guna booster apa.
Oh my goodness. Okay. Kalau you taruh itu maknanya you punya produk ni membunuh ayam. Okay, it's okay, it's okay.
Okay,
Tak apa,
okay.
nak suruh resultnya cantik. Okay. Ini is not recorded, right? Oh my goodness. Okay, tak apa. Oh my goodness. And another question is about the treatment with protease. Okay? It is actually to increase the digestibility.
Yeah.
Okay? Because you want to cut off the big molecule of proteins into monomers or dimers. Yeah.
Small peptide.
Yeah. So you need to, in your, okay, what is your problem statement actually? If you can briefly tell us your problem statement of your masters.
Okay, the problem statement is to yang digest itu. So to more digest actually. But today I tak tunjuk result digest tu sebab masih lagi dalam analisis.
Yeah, okay, so walaupun kita bagi protein tapi ayam tak boleh manfaatkan 100%.
Ya.
Disebabkan susah untuk digest. So you want to increase the digestibility by doing this enzymatic
Pre-treatment, by doing the pre-treatment.
in terms of economy, is that using this protease processing, is that economically viable for industry?
For the protease, industry. Okay, for industry, because the protease we only use maybe the small amount only. Maybe per ton tu sikit je, tak sampai kilo.
Did you do this economic analysis?
Tak termasuk lagi. Tak masuk lagi. Cost benefit tu tak masuk lagi. Tapi akan ada nanti. Last.
Is there any side effects of protease towards the chicken?
Okay, side effect protease toward the chicken, and as I show tadi earlier, the mortality rate, the group with the protease, only protease T2, the mortality rate only 7%, which is 2 died along the 35 day. So I think the protease did not affect. Maybe slightly affect. Dia macam sikit je dia affect. Sebab dekat mortality rate dia bagus.
What happened to the chicken? Cerik-berik je?
Dia tak cerik-berik. Dia tak cerik-berik. Dia tinja. Tinja dia macam biasa je.
Okay.
Tapi
how do you propose that the death? What happened to the chicken? Is it protease tu pergi makan dia punya usus sampai usus dia bocor?
Okay, actually tadi saya ada tunjuk yang saya ada blood collection. And then maybe we can blood collection and also the histopathology to check the disease.
Okay, in future you can do forensic investigation.
Bukan forensik.
Iyalah, you sembelih ayam tu, belah ayam tu, you check lah dia punya perut dia macam mana.
Postmortem.
Oh, definitely, yeah. Okay. Dr. Laila, tak boleh, I kena stop.
Any question? Okay, so Nur Ratira, so nice and interesting presentation.
Yeah.
Okay, actually you have 3 results, yeah? T1 or T3. So for your appearance, which one you will prefer for commercialization?
For the commercialization, I prefer T3. Maybe we can reduce on protease.
Yes.
And we can combine with the salt because the salt is more cheaper in producing ni lah cost benefit tu. The cost.
Okay.
Yeah.
So how about the growth then? T3, because T2 have the more higher weight, yeah? The weight of the chicken, I think.
Yeah, T3, the at the end of 35 day 2, about 1.0 1.08 kilo compared to only 300, range 300 for the untreated.
Okay,
So beza jauh, different.
Okay, untreated means you not given any of the commercial feed. Yeah, untreated means you not given any of your formulation one or your commercial.
The untreated and the treated, all the formulation from me, not include the commercialized, not combining.
Oh, but they actually just propose that you need that one. Okay,
Yeah,
to
yeah.
test with commercialization because
yeah.
end of the day you actually want to commercialize, as a
Yeah.
product. So you have to compare. Okay.
Okay.
Thank you.
Okay, thank you.
Very kind question from Dr. Laila.
Ada lagi ke?
Oh, no, no. Okay. No, thank you very much indeed, Dr. Atira. A very interesting project. That's why so many questions.
Okay, thank you.
Thank you. Okay, now we end our session, and then again, I would like to record my gratitudes to all presenters, Chan Ke Xin, Rival, Firas, and also Nurul Atira. And please do not forget to come for the closing ceremony at 4:40 p.m. Yeah. So I suppose that's all for our session, and I'll leave back to our chairperson.
Hello. We have reached to the end of physical oral presentation. Thank you, moderators and all presenters. We will have a short break. The closing ceremony will commence at 4:40 p.m. Thank you.